Hi, everyone. We're at the top of the hour, so we're going to get started with our webinar for today. My name is Dr. Christina Asa. I'm a scientific application specialist here at Fujifilm Visual Sonics in Toronto. I'm really excited to present to you our speaker for today, who is going to talk about molecular magnetic resonance imaging of alpha v-beta-3 integrin expression in tumors using ultrasound microbubbles. And our speaker is Vertika Pathak. Just a few notes about today's webinar. We are recording our webinar for today, so it will be made available after our presentation. All lines are muted for the duration of the webinar, so please use the Q&A button at the bottom of your Zoom panel throughout the presentation to ask any questions you might have. Questions will be answered at the end of the session, and we do expect our presentation to be about 20 to 30 minutes, leaving us 5 to 10 minutes for questions. Okay, so today's speaker is Vertika Pathak, who obtained her bachelor's from DSB College in India in 2010 and was awarded the prestigious Vice Chancellor's Silver Medal. In 2013, she completed her dual degree master's course in nanotechnology and nanoscience at Amity University, also in India. For her master's dissertation, she joined Dr. Justin Haynes's research group at the Center for Nanomedicine at Johns Hopkins University. And she worked on developing mucus-penetrating nanoparticles for treating lung and cervical cancer. In 2016, she joined Dr. Lammers and Dr. Kiesling's research group at the Institute for Experimental Molecular Imaging, RWTH University Hospital in Aachen, Germany, as a doctoral student, and she's funded by the prestigious DAAD scholarship. The primary focus of her work is on alleviating tumor-associated hypoxia using nano and microbubbles in ultrasound-assisted blood-brain barrier opening. In the past 4 years, she has contributed to 5 publications. Her career objectives are to lead her own research group to advance novel research and improve public welfare. So without any further ado, I'm going to pass things over to Vertika.
Thanks so much, Christina, for the kind introduction. Hello, everyone. Welcome to today's session. My name is Vertika Pathak, and I'm a PhD candidate at Experimental Molecular Imaging Aachen. And my PhD is all about utilizing multifunctional microbubbles for hybrid imaging applications and theranostic applications. I hope you can all see my slides. Today, I will be sharing with you my recent work in which we investigated the possibility of using microbubbles that are commercially used as ultrasound contrast agents for molecular MRI applications. This is the outline of today's talk. We will briefly discuss about tumor angiogenesis and why its detection is important. Then what is the current state of the art and challenges for angiogenesis detection? Why we used microbubbles for molecular MRI in this study, and then the results and discussion and so on. Let's start with a bit of introduction about angiogenesis, which is defined as the formation of new blood vessels from the pre-existing ones to meet the high demands of oxygen and nutrients for growing tumors, and is one of the most studied hallmarks of cancer. It is now well established that highly angiogenic tumors tend to be aggressive and have a high metastatic potential, which correlates with poor prognosis. Due to the highly dynamic nature of this process, assessing angiogenesis is crucial for patients undergoing anti-angiogenic therapy treatments. As these drugs target the molecular pathways that are involved in neoangiogenesis, these therapies require rigorous and constant drug versus dose response and efficacy monitoring. Current clinical methods for angiogenesis detection rely on histopathological evaluation that often demands recurrent biopsy measurements and does not aid in patient compliance. Imaging is also used in combination with biopsy sampling. However, an ongoing dilemma exists whether conventional imaging, for example, CT, ultrasound, and MRI, can truly and accurately assess tumor angiogenesis, as generally these techniques are not sensitive to early effects of anti-angiogenic therapies, which overall makes the identification of responding and non-responding patients very difficult. Moreover, these techniques are generally unable to differentiate between the true disease progression and pseudo-progression. To address this diagnostic dilemma, molecular imaging techniques were developed that directly assess the key angiogenic biomarkers in the tissue and thereby enables early detection of changes in pathophysiological processes at cellular and molecular level. For example, it was demonstrated in a number of preclinical studies that molecular ultrasound in combination with targeted microbubbles was superior to functional ultrasound in differentiating highly angiogenic and less angiogenic tumors. More recently, the first in human clinical trial on molecular ultrasound in patients with breast and ovarian cancers with KDR targeted microbubbles has shown that the technique is clinically feasible and safe, and that the KDR targeted microbubble signal, KDR targeted molecular ultrasound signal correlates well with histological findings. MRI is another powerful modality which is highly desirable for molecular imaging, but suffers from its inherently low sensitivity to commercially available contrast agents. For example, gadolinium-based low molecular weight contrast agents. To overcome the sensitivity issue, nanoparticles such as liposomes, micelles, dendrimers, encapsulating high amounts of gadolinium or iron-oxide nanoparticles were developed. But if we look from an imaging point of view, targeted imaging with nanoparticles is difficult, which is also illustrated in this diagram. Nanoparticles extravasate from the leaky tumor blood vessels and non-specifically accumulate in the tumor tissue. Moreover, nanoparticles are also strongly taken up by the cells of reticular endothelial system, which overall makes it difficult to identify the true target bound fraction. Considering these above points, we in this study maneuvered micron-sized particles that remain completely intravascular, thereby provide high specificity of detection, and loaded them with superparamagnetic iron oxide nanoparticles to enable MR-based detection. In this context, the location of spions at the air-liquid interface strongly enhances susceptibility effects, thus making the agent sensitively detectable in T2 star weighted MR images. But before coming to the study part, I would like to briefly introduce about microbubbles for viewers who are non-expert in the field. Microbubbles, as the name suggests, are micron-sized gas-filled particles with an average diameter of 1 to 5 microns. Their shell can be made of lipid, proteins, and polymers, which can be further decorated with drugs targeting ligands, as well as polyplexes, for example, which can be used for gene delivery. When injected in the body, these gas-filled particles vibrate, resonate, and re-emit the sound energy, and thereby provide high contrast between the blood and the tissue. Our institute has a dedicated research group that focuses on utilizing these particles for multimodal and theranostic applications. We have shown in our previous reports that these microbubbles can be used to deliver drugs locally to the tissue. USP-loaded microbubbles can be used to monitor blood-brain barrier opening, and by functionalizing them with the right targeting ligands, we can also employ them for ultrasound-based molecular imaging applications. Coming back to our study, we synthesized these particles based on already established one-step loading protocol, further functionalize them with cyclic RGD and RAD peptides via streptavidin biotin coupling, and further characterize them for their in vitro and ultrasound-based MR relaxation, characterize them for in vitro ultrasound and MR relaxation properties. Finally, the performance of these agents for multi-scale, multimodal molecular imaging was evaluated by monitoring their localization in 41 breast tumors using MRI and ultrasound. Coming on to the results part, here panel A shows the dynamic light scattering results of spions demonstrating a narrow number-based size distribution. Panel B shows the Coulter counter-based measurements of spion-loaded and unloaded microbubbles, indicating a relatively narrow size distribution with mean average between 1 to 3 microns. The panel below shows the TEM images of both spion-loaded and unloaded microbubbles, demonstrating high encapsulation of nanoparticles in the polymeric shell, whereas the unloaded microbubbles showed an empty shell. Then different concentrations of spion-loaded MB were embedded in gelatin phantoms, and MR imaging was performed. As shown in figure A and B, we observe a dependence of microbubble concentration on the MR relaxation properties, with 1 into 10 to the power 9 MB per ml concentration showing the strongest T2 and T2 star effect, whereas the lowest concentration showing almost no effect. And this increase was not found to be linear. Next, in vitro competitive binding experiments were performed using 41 cells. Figure A shows here the schematic illustration of the binding experiments. Figure B shows the MRI results, C and D shows the ultrasound results. In MRI, we observe significantly higher R2 values for cells incubated with RGD spion-MB compared to cells incubated with spion-MB only. In line with this, also ultrasound imaging revealed highest area percent fraction of the MB signal when cells were incubated with targeted MB compared to non-targeted microbubbles. And moreover, when excess-free RGD was added to the cells before adding these targeted microbubbles, both R2 and area percent decreased significantly. So yeah, these findings suggest that the binding of RGD spion-MB to alpha-5 beta-3 integrin receptors in vitro can be quantitatively assessed by ultrasound and MRI. Encouraged by our in vitro results, we explored our imaging strategy to assess alpha-5 beta-3 integrin expression in vivo using MRI in mice bearing subcutaneous tumors. Figure A and B shows the representative signal intensity over time curves from the dynamic susceptibility contrast MRI of animals injected with spion-MB and RGD spion-MB respectively. Here, the red dots represent the mean signal intensity within the tumor ROI at different time points, and the blue line represents the fitted curve over the 300 time points. As we can see here, the curve of the spion-MB initially drops and then quickly returns to the baseline, demonstrating MB passaging in the blood into tumor blood vessels. In contrast, after an initial decline in the decrease in the curve, the RGD spion-MB stays at a low signal level during the entire period of the scan, hereby demonstrating microbubble binding and retention in the tumor. Next, we performed semi-quantitative analysis of these curves and we extracted several parameters. Here, the peak enhancement is the maximum loss in signal intensity from the baseline intensity and the maximum signal intensity in the curve. The AUC represent the total area under the curve, and it shows the total amount of microbubbles in tumor blood vessels. Gaussian slope tells us about the entry of microbubbles in the tumor vasculature, and the late enhancements tells us about the total signal loss in the signal intensity during the washout phase of the microbubbles. These results interestingly demonstrate that a 7-fold higher AUC was observed for animals injected with RGD spion-MB compared to spion-MB. And this augmentation of local availability occurred despite the fact that similar amounts of microbubbles were injected in all 4 groups. Therefore, this difference between them might actually be the difference in the specific retention of the targeted microbubbles in the tumors. Likewise, the late enhancement was also found to be 3-fold higher in the RGD spion-MB group compared to spion-MB group. And this also points towards strong retention and continuous binding of targeted microbubbles to alpha-5 beta-3 integrin receptors, while the clearance of non-targeted microbubbles from the tumors occurs fairly shortly. Similarly, target binding was also confirmed by ultrasound by using the destruction replenishment method. In this method, we intravenously inject both types of microbubbles, and then we wait for 7 to 8 minutes till they bind to the receptors. And then by using a destructive pulse, we destroy both circulating and bound microbubbles in the imaging plane. By subtracting the post from pre-destructive mean intensity, we can quantify the target bound fraction. In line with this, here also, in line with the MRI results, here also, no obvious differences were found in the peak enhancement. However, highly significant differences were found in the change in acoustic intensity after the application of destructive pulse in the RGD SPION-MB group compared to the other three groups. Overall, these results also confirm a much stronger retention of targeted microbubbles compared to the other three groups. To summarize, we in this study demonstrated the feasibility of using targeted microbubbles for molecular MRI of tumor angiogenesis. In this context, their intravascular persistent and thus low unspecific background signal generation makes them attractive for molecular MRI and ultrasound applications. Moreover, our ultrasound results nicely supported our MRI results. From a translational point of view, our hybrid methodology may be highly useful for interventional procedures where the tumor region is initially depicted by whole-bodied MRI, and subsequently the ultrasound real-time feedback is used to guide tumor biopsy and excision at the organ scale. In addition, it might be interesting to evaluate these targeted SPION-MB for MR-guided sonoporation or sonopermeabilization of tumors, hereby taking advantage of the real-time multiparametric MRI feedback on the tumor tissue. Excuse me. Finally, I would like to thank both my supervisors who gave me the opportunity to work on these projects, all my co-authors that are involved in the study, of course, all my friends and colleagues at XME, my DAD scholarship, and last but not the least, Fujifilm Visual Sonics for giving me this nice platform to present my work. Thank you to all the viewers for joining today. If you have any comments or questions, I would be happy to take them.
Thank you so much, Vertika, for that excellent presentation. So at this time, we're going to open the floor to any questions. Again, I'd like to remind the audience, please use the Q&A button at the bottom of your Zoom panel in order to ask any questions to Vertika. So to kick things off, we have a couple of questions that have come in. The first has to do with next steps, more or less. Do you see any additional hurdles when integrating these nanoparticle-loaded microbubbles versus traditional microbubbles when trying to get this approved for the clinic or commercialized for diagnostic use in the clinic?
Yes, actually we use streptavidin biotin coupling, which cannot be used because when injected, these can be highly immunogenic. But we also have, because this is the PBCA based polymers, by hydrolyzing them, we have these free carboxylic groups. And we also can employ the EDC based coupling, which is approved for the clinic. So I particularly don't see it as a hurdle. It's just streptavidin biotin coupling is really strong and it was easy to use that method. However, that being said, we also have other conjugation methods that work really well.
Great, thank you so much. So the next question, I think you kind of asked or tailored a bit towards that last slide you presented. Do you see these microbubble, the specialized nanoparticle loaded bubbles being used to detect initial cancer or to monitor therapy in patients?
If we can use these targeted microbubbles, of course, they can be used for differentiating between malignant and benign lesions, as well as in case of anti-angiogenic therapies, they can be really helpful for identifying responders and non-responders, because these drugs do not elicit their response like chemotherapies. They are more, they indirectly elicit their response. So it's difficult to tell just by looking at a CT scan or an MRI scan whether the tumor is really growing, because sometimes a tumor can be highly angiogenic, but inside the cells can be dying. So I guess it can be used for both these purposes.
Okay, great. Since you did work with both ultrasound and MRI using these particles, did you perform any correlation analysis to show how the results compared when using these two different imaging modalities?
No, we did not show it. I did not show it yet in the webinar, but yes, this could be additionally done.
Okay, great. All right. Well, if there are no more questions, I think we're going to wrap up our presentation for today. And I'd like to thank Vertika very much for giving such a wonderful presentation. And if you do have any further questions that you think of after the fact, please contact us and we can get you in touch with Vertika. So I want to encourage you to please contact us using our website and our portal if you have any other questions pertaining to the webinar or any other imaging work that you're doing. Check on our website as well for on our webinars page for past recordings of previous webinars, as well as to register for our upcoming webinars. We have our next one is in March, and it will be a great presentation from a UPenn group on imaging of liver fibrosis. You can find us on all usual social media channels, and you can as well visit us at any upcoming conferences. And I just noticed right now, we do have an extra minute and a question just came through. So Vertika, you're not quite off the hook yet. I'll ask you this before we officially wrap up. But in vivo, many tissues and cells express integrin alpha v-beta-3. How do you ensure specificity with these bubbles?
That's a very good question. I would say we cannot ensure 100% specificity, to be honest, but we did perform the competitive binding using the same RGD, same cyclic RGD that we do have on our microbubbles, and we could see that the competitive binding worked really well. But of course, there are other integrins also in the tumors, I agree, and yeah, so it cannot be ensured. 100% specificity cannot be ensured with integrins, I would say.
Great, thank you. And thank you for the question. Our last slide here is just to remind you that if you do require any, I see the questions are starting to pour in a little later. So keep thinking you're off the hook, but you're not. So comparing ultrasound and MRI is one aspect. How about fusing the data and assessing the tumors using machine learning?
That is a very good question. I think these days it's really a hot topic, and we also have people working in our group that specifically work on these data learning algorithms. So it would be, of course, interesting to see this in future, how it can be fused and what parameters can be combined for this purpose. But as I said, this was not the focus of this study. In this study, we just wanted to show the first, the proof of concept of this methodology. So
Great.
yes, yeah, in future, it would be really interesting to see this.
Great. And the next question is, which kind of RGD?
So it's cyclic RGD-FK, arginine, glycine, aspartic acid, phenylalanine, and lysine.
Great, thank you. And did you test your contrast agent in a model of inflamed tissue?
Not yet in this study, I would say, but we have many papers that we have used these similar kind of microbubbles, polymeric PBCA based, for inflammation also, and have shown that the molecular imaging with ultrasound can be performed.
Great. All right. This is my last slide of, I guess, my long-winded conclusion here. So if there aren't any other questions, again, I want to thank Vertika so much for your presentation and answering these questions so gracefully. And thank you to everyone for attending our webinar today. And please have an excellent day.